Hi, I'm John Cooke. I'm professor and founding director of our Center for RNA Therapeutics here at Houston Methodist Hospital. So today we're going to be talking about RNA. You're familiar with messenger RNA. It is the transient signal from the nucleus of the cell to make a protein. So the way it works, of course, is the DNA within the nucleus, a portion of that will be transcribed into messenger RNA, which is then exported out into the cytoplasm. And in the cytoplasm, it's going to be translated by ribosomes into proteins. Now, we can therapeutically modulate that process by generating messenger RNA, encoding our protein of choice. And mRNA is like biological software. You can make any protein you want with the messenger RNA, encoding that protein. And the trick, of course, is to get it into the cell. And typically, we do that with lipid nanoparticles, LNPs, that can deliver the messenger RNA to the cell. And of course, you're familiar with vaccines, like the COVID vaccine, infectious disease vaccines, that provide the message to encode antigens that'll stimulate an immune response. So, white blood cells will come along and detect that antigen to trigger an immune response against that particular protein, which is, typically in a vaccine, it's a protein that is in that virus or that infectious pathogenic agent. But today we're not going to be talking about infectious disease. Today we're going to be talking about cancer vaccines, personalized cancer vaccines. So let me take you through that. So let's talk about the promise of personalized cancer vaccines. This paper in Nature this year got everyone's attention because what it shows us that personalized cancer vaccines can actually work in lethal diseases like pancreatic cancer. In this study, the investigators showed that patients that didn't respond to the vaccine had a poor recurrence-free survival of just over a year. Whereas patients that had a response to the cancer vaccine had an excellent recurrence-free survival over time, right here. Now we're planning to make our own personalized cancer vaccine at Houston Methodist, and we can do that because we have a center for RNA Therapeutics that has all of the elements necessary to generate novel RNA constructs. We start with our group that's doing RNA bioinformatics. So they can take a look through all of the RNA sequencing data that we have, the DNA sequencing data that we have, and help us plan our vaccines. And RNA innovation. We have RNA innovators who are making circular RNA and other forms of messenger RNA that are more stable. That construct design then is turned into a real RNA construct with our manufacturing team and facilities. We have clean rooms here and a GMP-trained team that can make clinical-grade RNA. Now we have, in addition to the RNA synthesis and purification procedures, we also have the ability to make lipid nanoparticles. So the lipid nanoparticles are basically the carrier for the cargo, for the RNA cargo, and are required to get the RNA into the target of interest, into the cells for generation of the protein. We have the assays to validate the integrity and purity of the RNA. These are FDA-required assays that help us to understand if the RNA is safe, if it's pure, and if it has the sequence that we desire. We can do the GLP preclinical testing here. We have a wonderful comparative medicine program. We have people helping us with the regulatory strategy, and we have a first-in-man clinical trials unit. So we have that whole cycle of activities that are necessary for generating RNA therapeutics. And we're going to be talking now about how we make our personalized cancer vaccine. So how do you make a personalized cancer vaccine? Well, it starts with the patient. Currently we have a IND approved by the FDA to treat triple-negative breast cancer patients. Triple-negative breast cancer is a bad disease, has a high recurrence rate. So once that tumor is resected, it's possible that it could occur. So we want to prevent that. So what we do is when the tumor comes out, we isolate the RNA from that tumor, and we isolate the DNA from that tumor. The analysis of the DNA tells us what mutations have occurred, because tumors mutate all the time. That's what makes them so dangerous. And we also look at the RNA, because that tells us what tumor proteins are being made, okay? That tells us what tumor proteins are being made. And we can look at the RNA, we can examine the RNA with DNA and RNA sequencing. And that information tells us where the mutations are in the DNA, and it tells us what RNA, tumor RNA is being made, and what tumor proteins are being made that are different from the rest of that person's body. That's what we're going to design the vaccine against. We're going to design the vaccine against these tumor proteins, because we don't want to trigger an immune response against the body. We want to trigger an immune response against the tumor proteins. So all that information gives us the bioinformatics that were required for selecting our neoantigen. That's a tumor protein that's different from the rest of the body. That neoantigen sequence is what we want to encode in our mRNA. So we encode the neoantigen sequence into our RNA, and we're able to make that RNA vaccine and then administer it to the patient. So that's how a personalized cancer vaccine is generated. That's currently ongoing, but let me tell you about the future. Let me tell you what's going to happen in the next 5 years. We've already talked about RNA. mRNA is biological software. Biological software. We can make that here at Houston Methodist. We can make the RNA. Now, there are several companies that are making boxes to synthesize mRNA. And if you have a box like this that can generate RNA, you don't need clean rooms. Now, we're lucky to have clean rooms here at Houston Methodist. We're lucky to have a GMP team and a GMP director. All of that here at Houston Methodist. And that's great. It allows us to be pioneers in the era of RNA therapeutics. But ultimately, that's not something that can be replicated around the country. We need something that can be replicated around the country that's going to make vaccines, cancer vaccines, affordable and accessible to everyone. So that's why I think these boxes are really interesting. This is the hardware. And one company is NTX Bio that's making this hardware. There's other companies that are interested in this area as well. But the idea would be you put the directions for generating the RNA, the directions we could provide in the form of a DNA template. So we can make that. And then that goes into the box. And the box has, also going into the box are all the reagents that you need, like nucleotides, to make the RNA. So you give the box the directions. The box has the nucleotides in it and the enzymes in it. You need enzymes too, and buffer. And this has a hollow fiber technology that generates the RNA, and then that generates the RNA. Then it has to be purified, and then out comes your drug substance. So the idea is that we have the hardware that's being built, and we got the software. Put those two together, and it's like Silicon Valley all over again. This is going to take off. This is going to be amazing, because we're going to have these boxes in every major medical center in the country developing personalized cancer vaccines and other personalized RNA therapeutics. We want to do it here first. So again, we're talking about the future here. And in the future of our program, here in the Texas Medical Center, we want to start diffusing this technology out to our other hospitals in the greater Houston area. The idea being that ultimately each of these centers will have a box. Now, we'll still need a hub for this program initially, because what will happen is that these hospitals will identify the patient, they'll send us the tumor for sequencing and neoantigen design, as I've been telling you, and then we can send back to them the DNA template to put in their box. The DNA template are the instructions for the personalized cancer vaccine for that patient. So the patient will be treated at the outlying hospital, their tumor will be sent to us for sequencing, and then we'll be sending back the DNA template to the box so that the personalized cancer vaccine can be made on site for that patient. So there's no need for the patient to travel, there's no need to ship things around other than the tumor itself for sequencing, and then the DNA template, which is very stable, and that'll be sent to the outlying hospital. So that's the future. It's very exciting because personalized cancer vaccines have great promise, and this ability to make a hospital-based RNA therapeutic is going to change the way drugs are developed and distributed. There'll be more hospital-based programs, university-based programs for personalized RNA therapeutics, including personalized cancer vaccines. That's going to make this exciting new technology affordable and accessible for all.